All right, well, hello everyone. So we're going to begin the process here of looking at something that's related to kind of the last major topic that we talked about being reproduction. We're going to talk about development, a little bit about pregnancy, and heredity. Okay, so this first part that we're going to look at is going to be focused on development and what we'll call embryological development, meaning the development of the embryo from the point of fertilization with the creation of a zygote to a set of cells called a morula, and then this progression and this further development of this kind of little structure, this collection of cells that will ultimately develop into what we'll recognize as an embryo, and then ultimately a fetus, right? So as we do this, I want you to think about something related to development, something that's related to pregnancy, heredity here, and that being what kind of drives all of this forward, right? So what is driving the the union of a male gamete to a female gamete, right? And again, I think of this at a very large scale, right? And so what this comes down to is this comes down to passing along genetic information, right? This comes along to contributing your genes, trying to get your genes into the next generation, right? And so if you think of it from an evolutionary perspective, right, there are certain genes, there are certain types of genetic information that are part of an individual, part of an animal that are better suited for specific environments that give them some advantage in their ecosystem, in their environment, which allows them to survive, which allows them to reproduce at a greater rate than an individual without that particular set of genes, right? And so it's this idea of natural selection, right? There's also this idea of sexual selection, right? So meaning that there are certain individuals, there are certain characteristics, there are certain features that are more attractive, they're more appealing to members of the opposite sex. And so if you have those features, then you're going to attract the opposite sex, you'll reproduce, and again, you will pass along your genetic information, right? So a very classic example of this is the peacock, right? So here you can see a male and a female peacock. So which one of these on the screen, which one of these is the male peacock and which one of these is the female peacock, right? So hopefully that you see the one on the right, right, with its big tail feathers, that's the male peacock, and then this other one without the illustrious, this nice big, tail feathers, that's the female. Okay, so in this case, the large tail feathers with these elaborate, patterns, it's very attractive to the female. It's attracting a mate for the male such that it allows the male to pass along its genetic information. So it breeds, passes along its genetic information, and hopefully that material, that genetic material, makes it into the next generation and it passes along its genetic information, right? And so that's how you can end up with these elaborate patterns, these elaborate phenotypic, physical characteristics, that is through, at least in part, through this idea of sexual selection, right? So what we're going to focus on is development. Okay, so this is kind of a general overview of what we're going to be looking at. So we're going to start with fertilization. Okay, so meaning the gametes that we've talked about previously, the male gamete, the sperm, and the female gamete, the oocyte, right? So what you see here, those are going to unite. They will fertilization will take place, and you form the first diploid cell. So notice here it says 2N. That 2N means diploid. Okay, if it's just N, like the sperm and egg, the sperm and oocyte, that's haploid. So we form our first diploid cell called a zygote. The two nuclei of the gametes, the sperm and the egg nucleus, they fuse. Now you have a diploid nucleus, meaning 23 pairs of chromosomes, or 46. And then that zygote undergoes this process called cleavage. Okay, and cleavage is essentially the doubling of the number of cells. So 1 to 2 to 4 to 8 to 16 to 32 to 64 to 128 and so on and so on. Right? So this idea of cleavage, we're dividing, we're increasing the number of cells. And then what we'll look at in later parts is what's happening to those cells as you're dividing, right? So as you go from 2 to 4, 4 to 8, 8 to 16 to 32 and so on, what's actually happening to those cells, right? They are increasing in number, but they're ultimately arranging themselves in a very particular way that's going to give rise to essentially all of your tissues. So think of all of our different tissues we've talked about: epithelial tissue, connective tissue, muscle tissue, bone, all these different things. It's going to give rise to all of those, right? We'll have a separate lecture, a separate video, and we'll talk about this process called gastrulation. I'll explain exactly what's happening there. We'll undergo something called folding, so the embryo will fold in 2 different ways, and then ultimately we'll move into the fetal period as well. So typically the first 8 weeks of development are categorized as the embryological period, or this embryonic development. This is categorized by the initial emergence of structures, the initial kind of differentiation of structures as well, so you start to see different types of things forming. And then the fetal period, which is after week 8, so week 9 essentially up to birth, is categorized largely by growth, right? So most of the structures of the fetus are there, or are formed by the end of the embryonic period, and then the fetal period is characterized by large-scale growth of those, right? Of the fetus itself, and then of the individual characteristics. So think arms, legs, fingers, facial features, things like that. Okay, so that's the fetal period, the remaining 30 weeks or so. Right. So if we start at the very beginning, right? If we start at fertilization. Okay, so fertilization, we're taking those haploid gametes that we've talked about previously, the sperm and the egg, you unite them together, and now you have your first diploid cell. So what is the first cell called? That is the zygote, right? So the zygote is the first diploid cell that is formed in the body. All of us, we started off as 2 haploid cells, if you want to think of it in this way. 2 haploid cells, a sperm and an egg, unite, and we all came from that initial single diploid zygote, right? So we grow, we develop, we undergo that process called cleavage that we'll look at, and again, we're going from 1 to 2 to 4 to 8 to 16 to 32 cells, right? All of this occurs, this initial fertilization period, and I'll show you some images here in a moment. This is occurring in the fallopian tube. Right, this is occurring in the fallopian tube, uterine tube, oviduct, whatever you'd like to call it, right? Typically the ampulla, all right? So remember that secondary oocyte is ovulated. It is swept into the uterine tube, the fallopian tube, and then usually somewhere in the ampulla, a sperm cell will meet it, right? Now, importantly, for fertilization, 1 sperm cell, okay? 1 sperm cell is going to fertilize under normal conditions, right? There are a set of events, there are certain things that occur to prevent multiple sperm cells from trying to fertilize, or from ultimately fertilizing an oocyte. So this process, if you have multiple sperm cells fertilizing a single oocyte, that's called polyspermy, right? Polyspermy, meaning mini sperm, right? So if you have 2 sperm, then this is called a triploid cell, or it produces a triploid cell, meaning 3 sets of chromosomes. So 1 from the oocyte, 1 from the first sperm, and 1 from the second sperm, right? You could also have a quadruploidy, or a quadruploid cell, meaning 1 oocyte and 3 sperm. All of these are not viable, okay? These will not develop into an embryo, fetus, and then ultimately birth. This is these triploid and quadruploid cells are not viable. So there's a sequence of events that occurs to prevent or block polyspermy, essentially making the oocyte impermeable to additional sperm cells that are attempting to fertilize it, right? Now, there's also a set of events that called capacitation, and capacitation is essentially the preparation of the sperm to be able to fertilize the oocyte. And this occurs once the sperm cells have entered the female reproductive tract, right? And so what this is essentially there are receptors on the sperm cells that bind progesterone, right? So remember, progesterone is a sex hormone, right? We typically associate it with females, and it binds to these sperm cells. It allows calcium to enter, and this stimulates the motility of the sperm, right? So they become motile, they become able to move independently, and they begin moving into one of the fallopian tubes, right? And in reality, they move into both. Okay, so some of the sperm go into the uterine tube where there's an oocyte, some of the sperm go into the uterine tube where there's not an oocyte, right? And so if the sperm that goes in, if it goes into the fallopian tube where there's no oocyte, then obviously that sperm is not going to be able to fertilize the oocyte, right? So that sperm will not contribute any of its genetic information to the zygote, right? The sperm cells that make it into the correct, if we want to use the term correct fallopian tube, right, meaning where the oocyte is actually present, then one of those will have a chance at fertilizing the oocyte, right? Now, this is something to think about. If it was any other sperm cell, okay, so we have an oocyte, okay, we have our one oocyte in the fallopian tube. We have our oocyte, we have our sperm cell coming from the male, working its way up through the vaginal canal, through the cervix, through the uterine lumen, and into the fallopian tube. If it was any other sperm cell that fertilized that oocyte, you wouldn't be here. Okay. If it was any other sperm cell that happened to fertilize that oocyte, if it was the sperm cell just to the left of the one that ultimately did, if it was that one, you wouldn't be here. Okay. So think about it in that case. Each individual sperm cell is contributing its unique genetic information. Okay, if it was any other one, you wouldn't be there, right? So think about that for a second. So these are the steps of fertilization. Okay, so you've got your sperm. It's coming in to the vaginal canal, cervix, uterine lumen, ultimately into the fallopian tube. It's going to encounter an oocyte, and it's going to penetrate that outer kind of layer of cells around the oocyte called the corona radiata. Right. So corona means sun, and then radiata meaning like these radiating fibers. So it's these cells, some of those granulosa cells that we've talked about previously, kind of extending out. That's the corona radiata. And one sperm cell, or really several, could penetrate the corona radiata, but one is going to undergo that acrosome reaction. Okay. So that, remember, the acrosome is that cap that kind of lines the outside of the head of the sperm. It's going to break down the granulosa cells. It's also going to break down the zona pellucida. The zona pellucida was that kind of thin, clear glycoprotein layer around the oocyte. And after that happens, essentially the zona pellucida around the remainder of the oocyte is going to harden. Okay, it's going to become very stiff, and what that's going to do is that's going to prevent any other sperm cells from penetrating it and contributing their genetic information. Okay, so again, remember, if you have that happen, meaning you have multiple sperm cells contribute their genetic information, that's called polyspermy, and that is fatal for the zygote, right? So it's fatal for that combination of sperm and egg. So it won't, it will not develop. Okay, so we penetrate our granulosa, our corona radiata. We go through the zona pellucida, it becomes firm and hard, and then the two nuclei, so notice here these are called pronuclei, a pronucleus. That's meaning the haploid nucleus, right? So you've got the pronucleus from the sperm, haploid, pronucleus from the oocyte, also haploid, those fuse, and now you've got diploid, right? So now you've got a diploid cell, okay? So that diploid cell is now 23 pairs of chromosomes, or 46, and that's the first diploid cell that's formed. That is the first diploid cell from which we develop from, right? So we develop after the union of these two cells. You call that a zygote. We develop from that diploid cell. Okay, so let's stop here. We'll pick up in our next part, and we'll begin talking about something called cleavage and the doubling of these cells, and then we'll ultimately get into what those cells turn into.